Hi, my name is Heather desJardins-Park. I'm an MD PhD student in the Longacre Lab. And I'd like to thank you for visiting my virtual poster for this year's Holman Day. So broadly, I'm interested in studying fibrosis, such as skin scarring and regeneration. And in this study, I'm specifically looking at an example of regeneration found in the animal kingdom, which is this MRL lab mouse strain. And this strain was kind of fortuitously discovered to actually regenerate ear punch wounds. As you can see here, this is an MRL mouse, and this is the ear punch wound when it's created. And then, starting about a month out, these wounds essentially completely close. And this is in contrast to other strains, which don't really heal such wounds to any appreciable extent. Now, this ear regeneration phenomenon is really interesting in and of itself, but on top of that, the regeneration of skin in MRL mice is actually a tissue-specific phenomenon. So we see that this regeneration occurs in ears, but notably in other areas of the skin, including the dorsum, wounds actually do not regenerate and instead form scars. So this is a really interesting kind of regenerative process with these really interesting features that we think we can leverage to better understand the mechanistic basis of regeneration. And to do so, we are relying on this method called DIFACE. Now, this method, which I'll explain in a bit more detail in just a moment, was pioneered by Hunter Fraser's lab in the biology department here at Stanford. And this whole project has really been a close collaboration with their lab. So this method called DIFACE has previously been applied to successfully identify genes underlying complex traits such as mating behaviors in non-mammalian species like Drosophila and fish, but it's never previously been applied to mammals. So we thought this was a really unique opportunity to see if we could use this novel method to determine what genes are involved in the ear regeneration trait that we see in these MRL mice. So again, as I sort of highlighted, these MRL mice have this really unique phenomenon of regenerating ear wounds. And we can actually see histologically as well as grossly that they are truly regenerating key tissue types such as cartilage and normal appearing dermis, in contrast to these cast ear wounds. And cast is just the wild type strain that we're using for comparison in this study. In pretty striking contrast, dorsal wounds in both MRL and other typical wild type strains like cast form these very fibrotic scars. And you can tell these are very, visually distinct from normal skin. They form these bare areas sort of grossly, and then histologically, you can see that there are no, hair follicles or glands that are seen in normal skin. These are not found in scars, and the scars have a much more dense collagenous matrix. So we then turn to this analysis called DIFACE, and the sort of key takeaway that I want you to understand about DIFACE is that we're looking for a very specific pattern of gene expression. So for the purposes of our DIFACE studies, we are using hybrid mice that have one MRL parent and one cast parent. And that's important because in those mice, for every gene, they're going to have one copy of that gene, one allele that is derived from the MRL parent, and one allele that's derived from the cast parent. And theoretically, there are variations that have developed over time to the MRL genome that are what's causing those differences in expression of some genes that are leading to ear regeneration just in that strain. And those variants don't exist in the cast genome. So due to these variants, obviously, there's something happening in ear wounds that is not happening in dorsal wounds that's leading to regeneration. And this can be detected as differential expression of those pro-regenerative genes, whatever they are, specifically in the ear wound context and specifically from the MRL allele. And you can see that depicted here as, this blue gene, this gene, which is the MRL version is in blue, the cast allele is in green, and you're specifically seeing increased expression just from the MRL allele, so not from the cast allele, and just in the ear, not in the dorsum. And that's because, again, we see this phenotypic divergence in the ear that is, very specific to MRL but we don't see divergence in the dorsum. MRL and CAST heal very similarly with regard to dorsal wounds. So this kind of pattern of expression is something that we are actually leveraging in our analysis. And this is based on the fact that from bulk RNA-seq data, again, in these F1 hybrid mice, so first generation, where one copy of each gene is from MRL, one copy is from CAST, we can actually look at individual RNA transcripts and assign which genome they came from, MRL or CAST, based on small sequence variants. And we can then look for genes that have the specific pattern of differential expression, which might be and likely include genes that are involved in driving ear regeneration. So just sort of broadly, when we quantified how many genes had this pattern of expression that we're interested in, we saw that many more genes exhibited that sort of, again, this pattern in the ear, which is shown in blue here, compared to the dorsum, which is shown in yellow. And that's just important because, as I said, the phenotypic divergence that we're interested in occurs in the ear and not in the dorsum. So this was sort of an important just kind of sanity check that this method is actually yielding results that are potentially functionally relevant in the process we're interested in. Further validating our methods, when we looked at fibroblasts, which are the ultimate mediators of fibrosis and scarring, we saw that the genes that exhibited this diphase pattern, which stands for differential allele specific expression, those genes were enriched for processes that we know are involved in wound healing, things like cell adhesion, as well as mutant phenotypes such as abnormal wound healing. So all this was really promising. And finally, kind of the last nail in the coffin was when we looked at a previous genomic study that it's something called a QTL study, or quantitative trait locus study. And essentially it identifies broad regions of the genome. So maybe, several thousand base pairs that are inherited in a manner that's statistically significantly associated with inheritance of a trait of interest. So in this case, ear regeneration. So these are regions of the genome that we kind of know pretty definitively have some genetic elements in them that are involved in the MRL ear regeneration phenotype. And when we looked at this previous study and integrated it with our results, we saw that there was significant overlap of our diphase genes with those regions. And this is really important because it pretty strongly suggests that we actually are finding functionally relevant genes. Now, one of the genes that overlapped with one of these QTL regions, so again, functionally relevant regions for MRL ear regeneration, was complement factor H, which is an inhibitor of the complement signaling pathway. And really interestingly, when we treated dorsal wounds in MRL, which again, typically form scars, when we treated those wounds with recombinant complement factor H, we actually saw a significant decrease in scar thickness. So this, I think, is really interesting, not only as a validation of this novel genetic analysis method, diphase, in mammals, sort of higher order organisms, but also we have sort of unveiled this potentially functionally and possibly therapeutically relevant finding of a gene and protein that may be able to induce pro-regenerative effects. So there's still a lot to study in this. Specific to the complement factor H finding, we want to further characterize these wounds that have been treated with factor H to see to what extent they're truly regenerating. So for example, analyzing for the presence of potential dermal appendages, which is one key differentiating factor between skin and scars, as well as analyzing the extracellular matrix to see to what extent it resembles a scar versus normal skin. But I think, this finding is really exciting for us, as our end goal is always, how can we reduce the burden of scarring to patients? And I think that future studies sort of based on our initial findings, both of complement factor H as well as the other genes that we identified with this diphase method, could unveil potential therapeutic sort of molecular targets to reduce scarring. Thank you very much for visiting my poster, and I'm happy to answer any questions during the poster session.